Okay, so this is the second recording of the neural cell development lesson. And in this part, we start talking about the neural crest. So neural crest are ectodermal cells that are formed during the neural tube development. If you remember, we mentioned those cells. Those are formed via epithelial to mesenchymal transition when the neural tube close up. So we have the neural plate, then they close up and form the neural tube. And the neural plate and the neural tube separate themselves from the external ectodermal cells that form the epidermis. In between the two, we have some ectodermal cells which then delaminate. We will see that in a minute. Those cells that delaminate from there and they move around are the neural crest. Now, from the point of view of evolution, the appearance during evolutionary time of neural crest is extremely important, because the evolution of neural crest, so the appearance of neural crest during development, permitted later on the evolution of important features like jaw formation and face and skull, as well as pigmentation and the development of the bilateral sensing ganglia in vertebrates. So from the evolutionary point of view, neural crests are fundamental, extremely important. So these neural crests are important in the development because they are responsible for the development of the peripheral nervous system, but also different elements of the skull and the bones of the face, for instance, as well as the pigmentation of the body. Those cells are produced along the anterior-posterior axis while the neural tube is formed. So in terms of what they do along the anterior-posterior axis, we can recognize for the neural crest different regions. So we have the cranial or cephalic neural crest, which are the ones that are found and migrate in the most anterior part of the developing neurons. In this cartoon, here we have a chicken embryo, so this cartoon represents a chicken embryo. The cranial or cephalic neural crest are responsible for the development of the face cartilage and therefore the bones. Remember that cartilage comes first and then bones come later, but also the neurons, the peripheral neurons associated with the muscles in the face, the pigmentation of the face, as well as connective tissue. A subpart of the cranial cephalic neural crest, which actually are found a bit posterior, are the cardiac neural crest. This cardiac neural crest are responsible for the development of melanocytes. Melanocytes are the cells of the body responsible for the pigmentation. Melanocytes are also called melanoblasts. So these melanocytes are distributed along the skin of people and they produce pigments. So these melanocytes produce more pigments with people that have darker skin and less pigment with people that have less darker skin. And they are also responsible for the production of more pigment when we tan, for instance. And they are distributed around the body. The cardiac neural crest are responsible for the formation of some melanocytes in that region. And the region we are talking about here is between somite 1 and 3. But they are also responsible for the development of peripheral neurons in this region, cartilage, therefore bones, connective tissue, muscles, and the muscles, especially the one around the large arteries. The trunk neural crest are responsible for the development of the dorsal root ganglia. So they are sensory neurons starting from somite number 7. They're responsible for the development of sympathetic ganglia neurons that connect the innervate the adrenal gland between somite 18 and 24, but they are also responsible for the development of the melanocyte of the belly and the dorsal part of the body, as well as the nerve clusters around the aorta. Then we have two groups of neural crest, the vagal neural crest, which are more anterior, and the sacral neural crest, which are very much posterior. But both of them are responsible to generate or to form, to develop the parasympathetic ganglia, which are the innervation of the gut. So why is that some neural crest can form bones while others don't? Well, this is due to expression of oxygens. So the more anterior neural crest, so the cranial and the cardiac neural crest, do not express oxygens, while the more posterior, the trunk neural crest, for example, do express oxygens, and therefore, because they express oxygen, they cannot, they are inhibited in producing cartilage and bones. From again an evolutionary point of view, this is quite interesting because if you think about prehistoric fish, a lot of prehistoric fish, their head, part of their body covered by bones. So they were like armored fish. And this is because this structure come from, were developed by neural crest cells. So it is reasonable to assume that normal evolutionary ancestral state of neural crest is to produce bones, and only later on during development some neural crests lose the ability to produce bones because in them oxygens have been activated. So how the neural crest cells, which are ectodermal cells, are specified and are distinguished compared to other ectodermal cells. So here is a simple cartoon representing an embryo, and we are looking at an embryo from the top. So here is the dorsal part, here is the ventral part of the developing embryo. So how the ectodermal cells become different kind of tissue? Well, ectodermal cells that are going to produce epidermis are exposed for a very long time to Wnt signaling and BMP signaling. So if these ectodermal cells are exposed continuously for a long period to Wnt and BMP signaling, they become epidermis. However, if pulse or short expression of Wnt followed by BMP are experienced by these cells become placodal cells. These placodal cells are responsible, for example, for the development of portion of the eyes. More posteriorly, if you want, cells that are exposed for a long period of Wnt signaling or Wnt signaling plus BMP become neural crest. And whether they're exposed to only Wnt signaling or Wnt signaling and BMP will determine which kind of neural crest they are producing. And in fact, we can have a look on the general neural crest gene regulatory network. For the neural crest, this regulatory network, so this network of gene expression that produce neural crest, start with Wnt and BMP signaling, as we can see here in this part of this picture. In the second part of this picture, this Wnt and BMP signaling induce expression of ectodermal transcription factor like MSX1. So this ectodermal transcription factor are expressed actually in most of ectodermal cells. MS6 now induce a neural plate border specifier like PAX3 and 7 and YEDX5 and 6. And these specifiers are present in both neural crest and dorsal neural tube. However, for the cells to become neural crest, they are going to express neural crest specifier, which is FOXD3, SOX9, snail, and SOX10. once the neural crest is specified, the neural crest is kind of multipotent and can become a different kind of cells. For example, the progenitor, the nearly born neural crest, has the ability as progenitorial cells to produce either cartilage, melanocyte, glia, and neurons. And what determines which fate these cells is going to have depends on endocrine factors outside the cells. So it depends what is the experience of the cells in their surrounding. They will determine whether the neural crest will become one or another tissue. For example, neural crest will produce cartilage are exposed to EDN1 and NPPC, while neural crests that become glial cells are exposed to neuroguanine and EDN3, and so goes on. So one of the important event in the life of a neural crest cells is what is called the delamination. We call delamination the separation of this neural crest from their original tissue. So the neural crest lose their adhesion protein to the ectodermal epithelium that is going to form either the epidermis or the neural tube and delaminate and move around. So they undergo what is called an epithelial to mesenchymal transition. So epithelial cells are cells that bind tight with each other, so they cannot move much. So this is the case of the epidermis cells, the ectodermal cells that become the epidermis, or the cells that become the neural tube. They bind tight to each other because they need to form a layer of tissue. On the other hand, the neural crest cells need to move around. They cannot be bind like in an epithelium. So they become mesenchymal. So they can delaminate and migrate toward their target regions. To drive this epithelial to mesenchymal transition are BMP and Wnt signaling, especially from the epidermis. But this BMP and Wnt signaling become efficient, or they can actually induce the epithelial to mesenchymal transition only when the level of noggin from the notochord and the surrounding somates is reduced. So, for example, the cells of the neural tube that are more ventral will never be able to become neural crest because the level of noggin is too high. While the cells that are far away, so near the apical part of the neural tube, can indeed become neural crest because of low level of noggin. One of the genes that is expressed by BMP is SNAIL2. So SNAIL2 is quite important. It forms a dorsal to ventral gradient. So there's high level of SNAIL2 in the near the dorsal part of the neural tube, while there is very low level of SNAIL2 toward the ventral part of the neural tube. SNAIL2, for example, inhibit SOX2. SOX2 is produced by the, ventral part of the neural tube. But another reason for SNAIL2 to be expressed is that SNAIL2 moderate and control the expression of adhesion protein, like N-cadherin, cadherin 6b, and E-cadherin. E-cadherin, if you remember, it is expressed in the ectodermal cells that become epidermis. N-cadherin is expressed in the ectodermal cells that become neural tube, while cadherin 6b is specifically expressed in the cells that are going to become neural crest. Now, SNAIL2 needs to inhibit cadherin 6b, so the neural crest cells can basically remove themselves from each other. So they are first initially they bind together because the expression occurred in 6b, but then snail to lose this binding so the neural crest cells can move around finally and separate themselves from the rest of the tissue. Now, wing signaling from the external ectoderm induced expression inside the now migrating neural crest of raw GTPase or raw A. Raw A induced expression of other genes inside the migrating neural crest cells, like FOXD3 and snail. But they also establish the expression of actin microfilament, and the actin microfilament, like we saw for the neurons migrating, the actin microfilament form protrusion in the neural crest cells toward the direction of travel of the neural crest cells. And the direction of travel is determined by the concentration of raw A inside the neural crest cells. So the neural crest, inside the neural crest cells, raw is more concentrated in one region and less concentrated in another region. And the cell will travel toward in the direction of less concentration of raw A. So, but where do these cells travel? So in this cartoon here, you have a nice representation of a developing vertebrate embryo. You can see the neural tube. Below the neural tube, we have the notochorda, the notochord. Below the notochord, we have the aorta. This is the anterior region, that's posterior region. Near the neural tube, we also recognize the sclerotum. So these are portion of the somite, so the sclerotum on the two sides, as well as the dermomyotome. So that you remember the somites are formed by the sclerotum plus the dermomyotome. And covering everything in this blue layer here is the epidermis. And in this cartoon, it looks like that we are opening the epidermis so we can see inside the direction of travel of the neural crest cells. Now, there are two directions of travel the neural crest cell can take: the ventral pathway and the dorsolateral pathway. The ventral pathway, in the ventral pathway, the cell travel through the sclerotum, through the anterior portion of the sclerotum, while in the dorsolateral pathway, the cell travel in between the dermomyotome, so in between the derma and the epidermis. Now, the most anterior cells the most anterior neural crestal cell travel to the ventral pathway because of expression of ephrin and slit that block migration through the dorsolateral pathway. So ephrin and slit are blocking initially the migration toward the dorsolateral pathway. So the more anterior and the earlier produced neural cells need to travel by the ventral pathway. And initially, some cells travel in between somites, and the cells that travel in between somites innervate, produce the neurons that innervate the aorta. However, very soon, expression of somatophorin 3F block that way, so the neural crest cannot travel anymore between somites, and they are forced to travel the ventral pathway, which is not in between somites, but through the somites, through the sclerotum. And the cells that travel to the sclerotum are going to become peripheral neurons. They are going to become neurons that innervate the adrenal gland. They are going to become glial cells, including Schwann cells. Now, here we are looking in this cartoon to the embryo from the top, and we see this orange structure here, this globular structure that are representing the different somites, and in particular different sclerotum. And we see that the anterior and posterior part of the sclerotum are represented by different colors. This is because the expression profile of the cells in the anterior portion of the sclerotum is different from the expression profile of the cells in the posterior portion of the sclerotum. The neural crest cells are inhibited to travel through the posterior portion of the sclerotum, and they can only travel through the anterior portion of the sclerotum, because the posterior portion of the sclerotum express somatophorin 3F and ephrins, which block the migration of the neural crest in this region. On the other hand, more posteriorly and later, the neural crest cell migrate through the dorsolateral pathway. They can do that because in these cells FOX3 is repressed, and therefore MITF is upregulated. MITF stands for melanoblast or melanocyte inducing transcription factor. So this neural crest cell that travel to the dorsal lateral pathway become melanocyte or melanoblast. So they are the cells that give pigmentation to the skin of the body. And it makes total sense because these cells are traveling in between the derma and the dermis. So then from that place, they can travel through the dermis and become the melanocyte that give pigmentation to the skin. So one of the things that MITF does is to induce a pathway in the cells to produce pigments. Another thing that it does, it induces the expression of epinephrine B2 and endothelin receptor, which bind to epinephrine and endothelin 3, and this binding allowed the neural crest cells to migrate to this pathway. So, for example, and this is important, epinephrines in the sclerotum was blocking the movement of the neural crest. But here now, because the neural crest are now expressing a different surface receptor, epinephrine actually allowed the movement of this neural crest in this region. Another thing that MITF does is to control apoptosis of the neural crest, or in particular in this case the melanoblast. The melanoblast are associated with pigmentation, and we have mutation in the animal kingdom, but also in humans, of MITF and other transcription factor, which change how often these melanocytes are killed by apoptosis. Apoptosis is programmed self-death. We are going to talk about this in a minute as well. So, for example, Dalmatian, the dogs, but also some breed of horses and some breed of other animals, they have the appearance that they have because of a mutation on the MITF pathway that cause random death of melanoblast or melanocyte. And this causes these kind of spotted phenotypes. The bones of the head are originated by both the neural crest and by mesodermal, the mesoderm of the head. But which bones originate from the mesoderm and which bones originate from the neural crest? Well, not so long ago, using the very same staining that you try to use in your practical laboratory, using beta-gal, Zhang and a collaborator were able to figure out which bones come from where. They use beta-gal under the control of Wnt1 which is a marker for neural crest, and they stain the bones of a mouse embryo, and they found that the bones that were stained with the colorimetric reaction in blue were the frontal and the interparietal bones. While when they used beta-GAL under control of MES, which is a marker for mesoderm, other bones were stained, and in particular were stained the parietal and the temporal bones. So they were able to figure out which bones originate from which tissue. So the frontal part of the head is a neuronal, derived from the neuronal crest. Now, BMP and Wnt signaling, we already mentioned that, coordinate the protrusion of the frontonasal and maxillary process of the head. So the BMP and Wnt signaling control the neural crest, and therefore also control the frontal part of the face, if you want, in humans, but other animals. FGF signaling from the endoderm also control different skeletal elements, but in particular FGF8 control the cranial neural crest proliferation. We'll see this actually in the next lesson. So there are a very small number of genes, including BMP, Wnt, and FGF8, that are controlling the shape of the face. Now, I don't know if you notice, but we look a lot like our parents. So small variation in the expression of these genes cause the appearance, so how we look like. These small variation are hereditary because we look like our parents. So these small variations are genetically encoded, and which change the shape of our face and therefore change our appearance.